Hello everyone, welcome to online biology lesson. My name is Ravi Kumar, biology teacher. In today's lesson, we are going to discuss about chemiosmotic hypothesis. Actually, what is this chemiosmotic hypothesis? In our previous videos, we discussed about light reactions. In photosynthesis process, plants they convert light energy into the chemical energy in the form of ATP and NADPH. Especially these light reactions are two different types. One is cyclic and another one is non-cyclic photophosphorylations. During the cyclic and non-cyclic photophosphorylations, we saw that the reactions produce the ATP. In those reactions, we saw while the energized electrons passing through different electron acceptors or carriers at cytochrome complex, the electrons lose their energy to become normal. That energy used in the synthesis of ATP means those cyclic and non-cyclic photophosphorylations they cannot synthesize directly ATP. There is another process which called as chemiosmotic hypothesis, which explain this process means the synthesis of ATP. Let us now try and understand how actually ATP is synthesized in chloroplasts. ATP synthesis is linked to the development of a proton gradient across a membrane. To understand, let's see this picture, and here inside this is the structure of chloroplast, and here this aqueous spot we call as a stroma, and this membranous system they form this grana and stroma lamellae. So this grana, this part we call as a granum. Each granum is made of a number of thylakoids. So what are the thylakoids here? Each thylakoid is disc in shape, and here you can see this is a thylakoid, and this thylakoid is double membraned disc shaped structure. Inside they have the space which is called as a lumen. Here you can see the lumen part. Especially non-cyclic photophosphorylation takes place on a membrane of thylakoid. In this process, like while the electrons are passing from one photosystem to another system, which causes the proton gradient across the membrane. Means, so this is a stroma part and this is a thylakoid, and here inside a lumen part. So there is a proton gradient occurs across the membrane. Membrane means here the movement of protons takes place from stroma to lumen. So this we call as a proton gradient. So due to these reactions, there is a movement of hydrogen ions or protons cause the across the membrane, which leads the proton gradient. So due to the proton gradient, the proton accumulation is towards the inside of the membrane. That means it's a lumen. Due to the proton movement, the proton accumulation takes place in the lumen part. So here let's try to understand what causes the proton gradient across the membrane. Why the protons move from stroma to lumen, or which reasons make the movement of protons from stroma to lumen. Let's see those reasons. The first reason is since hydrolysis of the water molecule on the inner side of membrane, the protons or hydrogen ions that are produced in hydrolysis accumulate the lumen of thylakoids. Let's see this. The first reason. To understand this first reason, let's discuss about this non-cyclic photophosphorylation briefly. In non-cyclic photophosphorylation, there is a series of photosystems which are participate. The first one is PS1 and second one is PS2. This non-cyclic photophosphorylation occurs on the membrane of thylakoid. This is a membrane of thylakoid. The non-cyclic photophosphorylation occurs on the membrane of thylakoid. In the process of non-cyclic photophosphorylation, first, photosystem 1 observed wavelength of energy 700 nanometers, and the reaction center, which is called as a chlorophyll A, release two energized electrons. So these energized electrons pass through different electron carriers and finally they reach NADP to form NADPH. Here, once the electrons released from the photosystem 1, finally they reach to the NADP to form NADPH. Means here PS1 is missing two electrons. These missing two electrons were replaced by PS2, photosystem 2. Here, photosystem 2 observed 680 nanometers of wavelength of light, and the reaction center chlorophyll A released two energized electrons. These two energized electrons again pass through different electron carriers. So while they are passing through the cytochrome complex, they lost their energy and become normal, finally reach to the photosystem 1 to replace the missing electrons. So when the PS1 gives the 2 electrons to PS1, so PS2 again missing 2 electrons here. These missing electrons are fulfilled by this water molecule. So in the presence of sunlight, this water molecule splits as 2 electrons, 2 hydrogen molecules or protons, and half oxygen molecule. So when water molecule splits as these compounds in the presence of sunlight, we called as photolysis or photolysis. What is photolysis? Splitting of a water molecule in the presence of sunlight as electrons, protons, and half oxygen molecule is called photolysis process or photolysis. This photolysis process occurs on the inner membrane of thylakoid. So here these electrons moves to the PS2 to fulfill the missing electrons here, and these protons pushed into the lumen part, and this half oxygen molecule wait for the another half oxygen molecule to form one complete molecule of oxygen. Then they escape from the this lumen part, and finally they go enter into the environment. So that's why plants, they are releasing or producing the oxygen in the process of photosynthesis. So this is the first reason why the proton gradient occurs across the membrane. Let's see the second reason. As electrons move through the photosystems, protons are transported or pumped across the membrane into lumen. Here, while the energized electrons from PS2 passing through the electron carriers near the cytochrome, as I told you, they lost the energy. This energy pumped or helps in the transport of protons from stroma to lumen, means this energy pumps the protons or hydrogen molecules from stroma to lumen part or matrix to lumen part. So this is the second reason why the proton accumulation takes place in the lumen part. And the third reason why this gradient takes place across the membrane. The NADP reductase enzyme is located on the stroma side of the membrane. Along with electrons that comes from the acceptor of electrons of PS1, protons are necessary for the reduction of NADP to NADPH plus H plus. These protons also removed from the stroma. Here the third reason is, once the electrons, energized electrons, comes out from the photosystem 1, these energized electrons finally they reach to the NADP to form NADPH. So for the reduction process, they also need the protons. So these protons, this NADPH to form NADPH plus. So they get the protons from the stroma, or they remove the protons from stroma to form NADPH plus H plus. So because of this reason, we can see the differentiation of the charge, the differentiation of this proton charge, the lumen part and the stroma part cause the difference. This we called as a proton gradient. In the lumen part, there is a lot of protons. Because of these two reasons, when they push here in this reaction, and this water molecule when they splits, they also push the protons from here to here. So due to this reason, inside of the lumen there is a proton accumulation takes place when you compare with the stroma or matrix. So this caused the proton gradient across the membrane. Above reasons make the proton decrease in stroma, while the lumen there is accumulation of protons. This creates a proton gradient across thylakoid membrane. Why we are or why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. How this gradient break down? The gradient is breakdown due to the movement of protons across the membrane to the stroma through transmembrane channel of the ATP synthase. Here you can see when the proton gradient occurs in the lumen part, now the protons wants to go back into the stroma. So this movement of protons from lumen to stroma occurs through a channel which is called as a ATP synthase. Here you can see this one is ATP synthase. ATP synthase enzyme consists of two parts. One is called CF0, is embedded in thylakoid membrane, and other portion called CF1 is produced on the outer surface of thylakoid membrane. Here, this is a ATP synthase. This is a CF0 part portion which is embedded in the membrane of this thylakoid, and this is a CF1 portion which is produced from the membrane. ATP synthase has a channel that allows diffusion of protons back the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes formation of ATP. Here, due to this proton gradient, the protons which are present in the lumen part goes back to the stroma or matrix area through this ATP synthase channel. While they're passing through this ATP synthase channel, they release enough energy to synthesize ATP here. The ATP which is produced along with NADPH will be used immediately in the biosynthesis of sugars in stroma. This is about the lesson today. I hope you understand the chemosynthetic hypothesis process or chemosynthetic process, which is very important in the synthesis of ATP. By knowing this, we can say that light reactions, they never produce ATP directly. The light reactions, they converts the light energy into the chemical energy in the form of ATP. So in this process, so there is a process which is called as a chemiosmotic, which is very important in the synthesis of ATP. For chemiosmotic process, what they need? Chemiosmosis require a membrane, a proton pump, a proton gradient, and ATP synthesis. So here you can see the membrane, proton pump, proton gradient, ATP synthase. Energy used to pump the protons across membrane to create gradient or high concentration of protons within the thylakoid lumen. ATP synthase has a channel that allows the diffusion of protons back across the membrane. This releases energy to activate ATP synthase enzyme that catalyze the formation of ATP. This is the brief information about chemiosmosis process. I think you understand very well about this one. Thank you very much. We are going to meet again with the new topic from the photosynthesis. Bye bye. Have a good day.